everybody. My name is Dr. Rebecca Borup. I am an anatomy and physiology teacher in Southern California. We're going to be talking today about molecular interactions. We will begin with a review of some basic chemistry and biomolecules and proceed from there to discuss pharmacology, how these molecules interact with each other and how we end up generating responses within the cell. Most of this is review, I hope, for many of you that you've already had a chemistry class. So we'll just begin with a conversation about molecules. A molecule is two or more atoms held together by a chemical bond. They can be, they're usually electrically neutral and may contain atoms of the same element or different elements. We can have very small molecules, such as this example here, where we can see that we've got some oxygen with just two atoms bound together. We have a little bit bigger with our methane, 4 atoms, bigger still with the tripeptide, 58 atoms, and then, for example, hemoglobin, where I don't know off the top of my head how many atoms there are, but certainly this is a collection. It's 4 proteins that have interacted with one another to form protein subunits interacting with one another to form the entire hemoglobin molecule. So they can be quite large. There are different chemical bonds that work to hold atoms together and to hold larger molecules together. We can break these chemical bonds down into the covalent molecules, covalent bonds, correction, which we have our nonpolar. Nonpolar are not soluble in water, and that's an important thing to remember. They're insoluble in water. And so we can call these lipophilic, lipid-loving, or we can call them hydrophobic. Polar molecules then are usually soluble. They tend to have partial charges that make them interact, or that allow them to interact with water quite well. And so these are typically known as hydrophilic. I suppose technically you could use the word lipophobic, but that's not usually used. I don't think I've ever heard it used. On the other hand, lipophilic is used relatively frequently, and I tend to use that term. Then we have our ionic bonds. Ionic bonds are formed when electrons are transferred and we end up with charged molecules. They are also very much hydrophilic. They dissolve quite well in water and they dissociate in water. We'll talk a little bit more about that. Then we have these hydrogen bonds. These are individually weak bonds, but collectively quite strong. DNA, for example, is the two polymers of nucleic acid are attached or linked to one another through these hydrogen bonds. Thousands and thousands of hydrogen bonds have become quite strong, creating a very stable DNA molecule. And then the last type of bonding we're going to talk about, it's not really a chemical bond so much as it is a relationship, and that's these intermolecular bonds. Typically we see these with large molecules, large proteins, DNA, other large molecules interacting with another large molecule, or alternatively with a smaller molecule. For example, going back to the hemoglobin we looked up at here, hemoglobin is quite a large molecule and it interacts with oxygen, oxygen binding at the center of each of these hemoglobin molecules. A little later in the semester we will be talking more detail about the hemoglobin, but these are essentially what we would call intermolecular interactions, with oxygen interacting with hemoglobin. This takes a quick look at our covalent bonds. I'm just briefly going to go over these. We've already talked about them, but we've got covalent bonds. The most important thing for you to remember is that we're sharing electrons, but not necessarily evenly. If we do play nice, we share them evenly, then we get a nonpolar covalent bond. And an example of these nonpolar covalent bonds, methane is a great example. The covalent bonds between carbon and hydrogen we're sharing electrons equally, neither one of them hogging those electrons, and so the molecule in total is nonpolar. On the other hand, we can have our polar molecules, and those are when we don't share things evenly. And when we have a molecule that hogs the electrons, we call that an electronegative molecule. And in this case, oxygen is very electronegative. And you can see here this symbol. Let me draw it a little larger so you can see it a little better. This symbol here refers to or represents a partial negative charge. It's not the same as a complete charge, such as we see in ionic molecules, but it's a partial negative charge. And then on the hydrogens, we have this partial positive charge. That's not supposed to be an S, sorry. A partial positive charge on the hydrogens. And so we set up a situation where we have these poles, hence the term polar. Ionic bonds, as I've mentioned, are a transfer of electrons. You may remember in your chemistry that we have different electron shells. The innermost shell has room for 2 electrons. As we proceed outward with different shells, and the larger, more complex the atom is, the more shells we have. But as we proceed outward, we end up being able to contain 8 electrons usually. And then this outer, let me switch quickly to a different color. This outermost shell is called these are called valence electrons. These are the electrons that tend to react with other molecules. And in ionic molecules, what we end up doing, so the shells are most stable when all 8 positions are occupied. And so if you look at chloride, for example, we have 1, 2, 3, 4, 5, 6, 7 positions that are occupied by chloride. That outer shell, those valence electrons, chloride has 7 of them that belong to chloride. But it really wants 8. Because 8 gives it more stability. So what chloride does is it snatches an electron from sodium. If we look at sodium, how many does sodium have on the outer shell? Only 1. It has exactly 1 valence electron. Now sodium, once again, prefers to have 8 in its outermost shell. So it could accomplish this goal by stealing 7 other electrons from somebody or some ion. But the easier way to get stability is to give up an electron. And once that electron is gone, if you look down here, we've got 1, 2, 3, 4, 5, 6, 7, 8. And the outer shell is empty. And so that gives sodium the stability that it needs. Chloride is also stable because this is the electron that it's stolen from sodium. And so this creates completely charged or fully charged molecules where we end up with sodium, chloride being negatively charged and sodium being positively charged. When these molecules are solids, they tend to interact with each other. You can see down here we get this box here represents a crystal. They often form crystals, such as a salt crystal that you likely have seen at your tabletop. And the chloride and the sodium are closely associated with each other because positive and negative charges attract to one another. However, when we dump these compounds into water, what we end up seeing is a process called dissociation. I want to distinguish between dissociation and dissolving. Dissolving refers to solubility in water. And molecules such as glucose dissolve very nicely in water, but the carbon and the hydrogen and the oxygen in glucose, so here's the chemical formula C6H12O6, all of these molecules are attached by covalent bonds. So even though glucose dissolves very nicely in water, it will not dissociate. So dissociation involves a process where these molecules, if I were to diagram sodium and chloride and split those apart, what ends up happening is that water acts to stabilize those positive and negative charges. So if you remember that water, oxygen has a partial negative charge, and so the sodium ion will be surrounded by water molecules, and those water molecules will orient themselves so that the negative, partially negative charges found on oxygen will associate or surround that sodium, creating a stable ion in solution. And so sodium will separate from chloride. Vice versa, if we look at chloride, we see something very similar, but in this case, instead of the oxygen associating with the chloride, we're going to have the hydrogens associate with the chloride, because remember, those hydrogens have the partial positive charge. Chloride has a negative charge, and so that partial positive charge will stabilize the chloride ion and allow it to be happy, if atoms could be happy, dissolved in water. And so the process of dissociation occurs when these ions actually separate from one another and dissolve and disperse throughout the liquid fluid. And in separating, they become fully and completely charged. Hydrogen bonds are normally, or the most common molecule that forms hydrogen bonds are water. And you can look up here, if we look at this example here, what we end up having is we see the partial negative charge of the oxygen on one water molecule being attracted to the partial positive charge on a hydrogen ion. And so we end up with multiple hydrogen bonds being formed, and this actually creates surface tension. And we can refer to these sometimes as hydrostatic forces. You'll hear me talk about hydrostatic forces later on in the semester. The surface tension here, if you look at a pond, for example, you can get little bugs, forgive my bad art. Water striders, for example, walking across the water, and it's because the surface tension keeps the molecules connected to one another so that they create resistance that the bug can use to walk on. Likewise, if you were to pour water onto a tabletop, you would see it beat up like so. And again, that's due to the surface tension created by these hydrogen bonds. Alcohol, on the other hand, doesn't form hydrogen bonds very well, and so if I were to dump alcohol, I guess I'll use a darker color, onto my tabletop surface, it would just essentially spread out all across the table until it very rapidly evaporates, which, by the way, hydrostatic bonds, these hydrogen bonds, also affect evaporation. Alcohol evaporates quickly, whereas water is a little bit slower. It takes a little bit more energy. You likely discussed that in your biology class when you talked about some of the characteristics of water that make life possible. The fact that water needs more energy to evaporate actually means it can absorb a lot of heat. And so areas that are near large bodies of water tend to be a little cooler because the water is absorbing that heat and taking it out of the atmosphere. There are other molecules that can form hydrogen bonds. One of the most common that we're going to see in physiology is nitrogen. Nitrogen will form these hydrogen bonds as well, and that's actually very important for DNA. For example, the hydrogen bonds that form during DNA are usually formed between nitrogens and hydrogens. Then we have our intermolecular bonds. These mean inter means between, whereas intra means inside. So an intermolecular bond is between two or more molecules. And those molecules can be large, such as with DNA, interacting with, one nucleic acid from DNA, one polymer interacting with the other polymer in DNA to form the double helix that we associate with the DNA that is the DNA structure. There are more than just hydrogen bonds, so we also have hydrophobic interactions, for example. So if you have a hydrophobic molecule that wants to hide from the water, often you'll see those molecules tuck themselves away inside a protein, for example. and associate with other hydrophobic amino acids inside that protein, and tuck themselves away.